Hi guys, welcome to TMZ channel. This is the second biochemistry lecture, Lipolysis. In this lecture, we are going to understand how to mobilize the fats outside of their stores and how to be oxidized to get energy. Here is a brief introduction about lipolysis. Where it occurs inside the cytoplasm, it is a catabolic process, means we are going to break down a large molecule into smaller units. The large molecule is tagged and the final products or the smaller units of fatty acids and glycerol. Rate limiting enzyme, hormonal control are going to be discussed later on during this episode. Before we start, we need to know a single detail about the stores of fats, white adipose tissue and healthy liver. Inside the white adipose tissue, the fats are stored in anhydrous form. They are anhydrous because they do not contain water. In a healthy liver, very little amount of tags are found or are stored. Instead, most of the fats are exported, packed with other lipids and apoproteins to form particles made up of lipoproteins. Lipoprotein particles are going to be known as very low density lipoproteins. These are synthesized inside the liver. The newly formed very low density lipoproteins are going to be secreted into the blood where they mature and start to do their function. The function is to deliver endogenous derived lipids to the peripheral tissues. By the way, endogenous means synthesized inside the body. So let's start fat mobilization and fatty acid oxidation. Fatty acids are stored inside the adipose tissue in form of neutral tags. These are the body's major fuel storage because they are highly reduced and largely anhydrous. Anhydrous do not contain water. Highly reduced because of the long CH chain. Fatty acid release from fat. Before reading the paragraph, let's draw a single concept map to understand this paragraph. Tag, dag, mag. Triglyceride, di, mono. Tag is made up of a glycerol attached to 3 fatty acids. Dag is a glycerol attached to di or 2 fatty acids. Mag is a glycerol attached to a single fatty acid. Here is the first substrate. To begin the process of the lipolysis, we need to convert tag to dag through adipose triglyceride lipase enzyme, and this is the first lipase to be used. Known as lipase because it breaks down lipids. Adipose triglyceride lipase. Then, to convert dag into mag, we need a very important and rate limiting enzyme, HSL or hormone sensitive lipase, and it's known as hormone sensitive because it is regulated via hormones. And finally, the mag is going to be acted upon by the mag lipase to get free fatty acid and free glycerol. This is the first thing in our lecture, which is fat mobilization. How we are going to get fatty acids and glycerol out of the tag is very simple in 2 steps. Tag into dag, dag into mag, and finally MAG into fatty acids and cholesterol. However, as anything inside the human body, this process needs to be regulated. Here is our cell, and here is the receptor. When adrenaline gets attached to the receptor, it activates adenyl cyclase enzyme, which converts the ATP into cyclic AMP. Cyclic AMP activate a cyclic AMP dependent protein kinase enzyme. As you know, kinases function is to phosphorylate other enzymes. So this protein kinase is going to phosphorylate HSL. By the way, HSL becomes activated when phosphorylated. And this is the first step to regulate HSL, is the activation. However, it needs to be inhibited as well. So all this is going to be inhibited when insulin attaches to the receptor. Here is what we have discussed now. The process of lipolysis is achieved by lipases. The first lipase is adipose triglyceride lipase, where we get DAG out of the tag. Then hormone-sensitive lipase is going to work on DAG to form MAG, and then finally MAG lipase acts on the MAG to get free fatty acids and cholesterol. This process is regulated through hormone-sensitive lipase. As we said, HSL is activated when phosphorylated. When catecholamines such as epinephrine binds to the receptors of the cell, as we said, adenyl cyclase becomes activated to turn ATP to cyclic AMP, and finally cyclic AMP activate proteins kinase. That's why it's known as cyclic AMP dependent protein kinase, because it becomes activated because of cyclic AMP. Because cyclic AMP is a sign of low energy, and we are already breaking down the fats to get energy. So this seems to be logic. On the other hand, insulin inhibits all this when binds to the receptors, since it leads to dephosphorylation of HCl, and as we said before, it's activated when it's phosphorylated and inactivated when dephosphorylated. Of course, epinephrine is not the only hormone controlling HSL. Others include norepinephrine, growth hormone, thyroid hormone, and other hormones which are not included here. Now we need to know the fate of cholesterol and the fate of fatty acids. Fate of cholesterol. Before reading the paragraph, we are going to discuss it in an easy handwritten concept map, and then we are going to come here. Do you remember the adipocyte where the MAG was turned into fatty acid and cholesterol? Unfortunately, this adipocyte does not contain kinase, which is important for phosphorylation. And so this adipocyte is unable to metabolize the cholesterol. So we need to ask for help. Here is the organ which helps others. Liver. Sorry for the bad drawing. liver do have kinase. So the glycerol produced inside the adipocyte is going to be transported through the blood to reach the liver here. Once it digests the liver, the kinase works on the glycerol, and we get glycerol 3-phosphate. Okay, how we are going to use the glycerol 3-phosphate? This glycerol 3-phosphate is going to be converted into DHAP, and I hope you remember it. DHAP may be used either for glycolysis or gluconeogenesis. However, this is not the only fate of glycerol 3-phosphate. Actually, it may be used to resynthesize a tag, which would be stored inside the liver. Here is what we said. Glycerol released during tag degradation cannot be metabolized by adipocytes because they do lack glycerol kinase. Instead, glycerol is going to be transported through the liver, through the blood to reach the liver or sometimes kidney, to be phosphorylated there through the kinase found there. Inside the liver, the glycerol 3-phosphate either be used to form a tag or converted to DHAP, which is dihydroxyacetone phosphate, which is then converted to glyceraldehyde 3-phosphate. I hope you remember these, and if you do not, please return to previous episodes for the previous modules. Okay, glycerol 3-aldehyde 3-phosphate is going to either rejoin glycolysis or gluconeogenesis. And now the first product, which is glycerol, is done. Actually, this figure shows you how the phosphorylation occurs, if you need it. What is the fate of the fatty acids then? The fatty acids are going to be transported through the blood, but actually they are not free fatty acids within the vessel. They need a carrier, like a taxi. Our taxi here is albumin. Having a taxi, which is albumin, until we reach whatever tissue that requires fatty acids: skeletal muscle, heart muscle, renal cortex, anything. Once they get inside the tissue, fatty acids are going to be activated. Being activated means they are going to turn into CoA derivatives. They are going to be known as fatty acetyl-CoA, so that they will be oxidized to get energy. Oxidation occurs inside mitochondria. Why? To be as near as possible for electron transport chain, if you remember. And if you do not remember, etc., go for the previous episodes for locomotor module. There's an important hint you need to know, that RBCs cannot use fatty acid as a fuel. Actually, because they do not contain mitochondria. They only use glucose. Brain cells do have mitochondria, but it did not use fatty acids for energy. Actually, we do not know the reasons. Okay, now the fatty acids are found within the cytosol in their new form, which is CoA derivative. But here is a surprise. We are going to face a problem. that the membranes of the mitochondria are impermeable to CoA. Before going through this part, we are going to draw a single map to understand it. Now the fatty acids, or especially longer chain fatty acids, have reached the cytosol, where they are activated to form their CoA derivative. By the way, this occurs by the help of thiokinase enzyme or acetyl-CoA synthetase. And now we have fatty acid acyl-CoA. The problem with it that the mitochondria does not allow CoA to pass through it, so we need a special carrier. Here is an outer membrane enzyme, another outer membrane enzyme, and inner membrane enzyme. I'm going to tell what are these. This is known as CAT1. Now CoA or acyl group cannot pass through, so we need help. We have a special carrier. This special carrier is known as carnitine. Okay, come carnitine, help us. Okay, give me your acyl group. Okay, the fatty acid is going to give the acyl group to the one who's known as carnitine. This occurs by the help of this enzyme, which is known CAT1. The product of this step is acyl carnitine, a carnitine who's carrying the acyl group of the fatty acid, and free fatty CoA. Now the free fatty CoA could get easily inside, and the acyl carnitine sadly stands outside. Okay, now how to get inside? Don't worry, there is a translocase specially for carnitine. Whatever carnitine carries, even acyl, he can get into. Okay, it gets through this translocase. However, there must be a free carnitine at the same time through this translocase to get outside, to allow this carnitine who's carrying the acyl group to get inside. Okay, now we are inside. Who's there? A free fatty CoA inside the matrix, as well as the acyl carnitine. Now the fatty acid is telling the acyl carnitine, give me my acyl group. So we need another enzyme to help for this step, an inner membrane enzyme known as CAT2. Now carnitine, please give the acyl group to the fatty acid so that everything turns back to normal. So we get fatty acyl CoA plus carnitine. I hope this helped you. After long chain fatty acid enters a cell, it's converted inside the cytosol into its CoA derivative by longer chain fatty acid acetyl-CoA synthetase, or briefly thiokinase. Beta oxidation occurs inside the mitochondrial matrix, and so we need to cross the inner mitochondrial membrane, which is impermeable to CoA. So we need a specialized carrier to transport longer chain acyl group from the cytosol to the mitochondrial matrix. Remember, the carrier is known as carnitine, and this is a rate-limiting transport process known as carnitine shuttle, and it's a rate-limiting actually because it can be regulated. This is how activation or how addition of acyl-CoA was done. And here is the translocation steps. As we said, the acyl group is transferred from the CoA from the fatty acid to the carnitine by CAT1, or sometimes known as CPT1, which is carnitine palmitotransferase 1. This is an outer mitochondrial membrane enzyme. Second, the acyl carnitine is transported through the membranes into the mitochondrial matrix by the exchange of free carnitine, as we said. He's going to enter the matrix, and at the same time, a free carnitine is going to go outside using the same translocase, which is known as carnitine acylcarnitine translocase. It's also known as antiporter, and this is an enzyme of the inner mitochondrial membrane. Now, carnitine palmitotransferase 2 or CPT2 or CAT2 is going to work. This is an inner mitochondrial membrane enzyme. This enzyme function is to return everything back to normal. And so, this step or this content must be equal to this. Before entering, we had free carnitine, fatty acyl-CoA. So inside the matrix, we must have the same thing, fatty acyl-CoA and carnitine. If this does not happen, so there is a disease. CAT2 catalyzes the transfer of acetyl group from carnitine to CoA inside the mitochondrial matrix, regenerating free carnitine and returning the acyl group back to its fatty acid. As anything in our body, such a step needs regulation, because if it doesn't go well, problems would happen. How to inhabit CAT1? Why to inhabit CAT1? Because I don't want the longer-chain fatty acids to enter inside the mitochondrial matrix, actually because I don't want to break it down or to get energy. Now I need to use it to form tags instead. So I need to inhibit the enzyme, helps fatty acids to get inside. This occurs by malonyl-CoA. Remember, this is an intermediate in lipogenesis, which was used in the first lecture in this module. So it prevents the entry of long-chain acyl groups into mitochondrial matrix. Therefore, when fatty acid synthesis is occurring inside the cytosol, which is indicated, of course, by the presence of malonyl-CoA, the newly made palmitate is not transferred into mitochondria and not degraded. And so, if we do not need energy, this shuttle is going to be inhibited in order not to waste our storage. Carnitine sources. We are going to talk how our hero carnitine have helped the fatty acid to get into the mitochondria. But who's carnitine? How did it get into their mitochondria? Actually, there are two ways to allow carnitine to get into our body. Easier through diet, and mainly this occurs through meat products. When you eat meat products, you are getting carnitine. And it synthesis from particular amino acids, lysine and methionine. Actually, this occurs inside the liver and kidney cells and does not occur inside skeletal or cardiac muscle cells as any enzyme or as any carrier or anything else in our body. Deficiencies may occur. Any deficiencies in carnitine leads to decreased ability to use our long-chain fatty acids as metabolic fuel. Primary carnitine deficiency is caused by defects in a membrane transporter that prevent uptake of carnitine by cardiac or skeletal muscles and kidneys. Because cardiac and skeletal muscles mainly cannot synthesize carnitine, so they are taken out from diet. So we cannot take it up into the cell. Where can it go? Excreted in the urine, because it's not used. And now we cannot synthesize or metabolize the long-chain fatty acids. Of course, there is a solution, don't worry. Treatment. The treatment includes carnitine supplementation. Secondary, defects in the fatty acid oxidation itself, which we are going to go through later on during this episode, lead to accumulation of acyl carnitine because defects inside the mitochondrial matrix during fatty acid oxidation would make it difficult for the acyl carnitine to enter the matrix, so it's going to accumulate outside in the cytosol. And of course, the cell doesn't want this to happen, and this may lead to injury, so they are going to be excreted in the urine, unfortunately. I'm saying unfortunately because acyl carnitine is important for regeneration of carnitine, and such a thing is going to decrease carnitine availability. Acquired secondary carnitine deficiencies may be due to liver disease. Actually, this is acquired because the deficiency maybe is not congenital, and liver diseases such as infections or injuries, fibrosis, cirrhosis, however, most commonly occurs later in life. As you know, the liver is the factory where carnitine is synthesized, so any liver disease or injury to hepatocytes lead to decreased carnitine synthesis. In addition, anti-seizure drugs, who taking these drugs are going to have decreased renal reabsorption. When we are going to go through the renal filtration, you are going to know that everything is filtrated out and then reabsorbed back. When renal reabsorption is decreased, what we have thrown is not getting back, so we are going to lose everything. That's why these drugs decrease the carnitine availability. Okay, we have finished the problem. Now, the longer chain fatty acid is there. How's going to be oxidized to get energy? Remember, till now we didn't oxidize the fatty acid. Actually, oxidation occurs through cycles, about 4 cycles. This one is the first cycle. It consists of 4 steps. The 4 steps are dehydrogenation, hydration, another dehydrogenation, and finally cleavage. Dehydrogenation is equivalent to oxidation, which is the removal of hydrogen. It occurs at carbon number 3 and carbon number 2. We are going to remove an H and another H here, so that fad turns into fat H2. Fat H2 will be used for generation of ATP through ETC. ETC, please refer to the previous episodes. This occurs by an enzyme known as acyl-CoA dehydrogenase. Then addition of water to carbon number 3 and carbon number 2 occurs, and this occurs via the help of enoyl-CoA hydratase. The third step includes another oxidation or dehydrogenation by 3-hydroxyacetyl-CoA dehydrogenase. And finally, we are going to cleave or to cut or to remove 2 carbon atoms in the form of acetyl-CoA by thiolase. Thiolase is the enzyme responsible for this step. To get fatty acyl-CoA and acetyl-CoA, here are 2 carbon atoms removed. Here's an important note. We are going to remove the carbons from the carboxyl end, and we start the reaction by the beta carbon, which is number 3. That's why it's known as beta oxidation reaction. After all these, look here, we turned the fat into fat H2. For the second dehydrogenation or oxidation reaction, we turned the NAD into NADH plus H. So this NAD and fat H2 will be used inside the electron chain reaction to get ATP. So we need to calculate energy yielding from beta oxidation. The oxidation of a single molecule of palmitoyl CoA to oxygen, carbon dioxide and water produces 8-acetyl-CoA, 7 NADH and fat H2, which gave me a total of 131 ATP to be generated. However, the activation of fatty acids requires 2 ATPs, so the net yield is 129 ATPs. How it was calculated? You know that NADH produces, a single NADH produces 3 ATP inside the ATC, and the fat H2 produces only 2 ATPs. What is new in this lecture that acetyl-CoA produces 12 ATP inside the Krebs cycle. Eight acetyl-CoA multiplied 12, 96. 7 NADH produced there, multiplied by the 3, since single NADH produces 3, so 3 multiplied by 7, 21, and 7 fat H2 multiplied by 2 gives me 14. Add all these results, give me 29, sorry. Again, this step is regulated. The first regulatory signal is the level of ATP inside the cell. If the ATP is high, the rate of the beta oxidation will decrease because we do not need energy. So, no need to exhaust the cell producing energy. And as we said before, malonyl-CoA, which is a precursor for fatty acid synthesis, inhibits CAT1. And since CAT1 is inhibited, fatty acids cannot get into the matrix, and so no beta oxidation occurs. This means that it inhibits beta oxidation. During fasting, oxaloacetate is depleted inside the liver due to gluconeogenesis, and this impedes the entry of acetyl-CoA into Krebs cycle, instead of using it for beta oxidation. And causing it to be converted into ketone bodies. Ketone bodies will be discussed on the next episode, don't worry. However, all this lead to inhibition of fatty acid beta oxidation. And so during fasting, no fatty acid beta oxidation occurs, and instead we need gluconeogenesis. This is a table of comparison just to summarize all of the above. For example, beta oxidation. As I said before, beta oxidation occurs for longer chain fatty acids. But there's something known as very long chain fatty acids, or those 22 carbons longer. First, these fatty acids are going to be oxidized inside peroxisomes instead of the mitochondria. After repeated cleavage, the very long chain fatty acids are going to turn into longer chain fatty acids, which are now available to be linked for carnitine and go through the previous steps. Unsaturated fatty acids are slightly complicated more than the saturated ones, but actually they give less energy because they are non-saturated and they are less reduced than the saturated ones. As you know, we are removing 2 carbon atoms in the final step in the beta oxidation because this is an even numbered chain fatty acid. What if it is odd, containing odd number of carbons? We are going to use the same mechanism. Instead, we are going to remove 3 carbon atoms in the final step. This is the only difference, or this is what the paragraph is about. Fat burns on flame of carbohydrate. Remember that when fats are broken down into fatty acids and cholesterol, they are oxidized via beta oxidation, this is the fatty acids, and glycolysis for the glycerol to produce acetyl-CoA. For acetyl-CoA to be oxidized, TCA cycle is there, and oxaloacetate is formed from pyruvate by carboxylation. And this supports and maintains the TCA. And so the glucose is the primary source of the oxaloacetate. So that through oxaloacetate, carbohydrate fuels the TCA cycle to continue the flame of the acetyl-CoA. I hope you get it. Again, as we have broken down the fat, the final product was acetyl-CoA. For this acetyl-CoA needs to be broken down or oxidized, we need to maintain the TCA cycle. For the TCA cycle to be maintained, oxaloacetate needs to be there. Oxaloacetate actually is formed by the carbohydrate. Because carbohydrate give me pyruvate, and pyruvate carboxylation leads to oxaloacetate. So as soon as glucose is there, fatty acids breakdown or fat breakdown would be continued and maintained. This is what fat burns on flame of carbohydrate means. Peroxisomal alpha oxidation. As we said, beta oxidation because we start with the third carbon or with the beta carbon. Now we are going to start with the alpha carbon, which is carbon number 2. This occurs with particular fatty acid, phytanic acid, which is branched chained, which is found in dairy products and meat. Actually, it is not a substrate for acyl-CoA dehydrogenase. It goes through 3 steps: dehydroxylation, oxidation, and decarboxylation. It does not require CoA and does not generate high energy phosphates actually. It does not require CoA because it does not need to be activated because of the methyl group already in its beta carbon. So no need to activate the beta carbon. That's all what you need to know about the peroxisomal alpha oxidation. Omega oxidation pathway is a minor pathway that occurs inside the endoplasmic reticulum of many tissues rather than mitochondria. This process occurs with medium-chained fatty acids of adipose tissue. Remember, there is a great difference between oxidation of long-chain fatty acids, very long-chain fatty acids, which was done by the peroximes, or medium-chain fatty acids, which occurs here by endoplasmic reticulum. And finally, the clinical part of the lecture, defects in fatty acid oxidation. What if all of the above steps didn't go well? The first deficiency is in carnitine. As we said, either the liver is not working well, there is decreased renal reabsorption, and we have already mentioned the causes. This leads to inability of the mitochondria to oxidize fatty acids because actually fatty acids was not allowed into the matrix. The treatment here is oral carnitine administration. Then the second problem is CAT1 deficiency. This deficiency results in recurrent muscle pain because muscles have no energy. What if the glycogen stored inside the muscle is depleted and we cannot oxidize the fatty acids? We need to contract and relax and there is no energy, so pain and fatigue would occur inside the muscles. Here's an important thing in this paragraph. You need to know that carnitine acyltransferases, which are the translocase enzyme, if you remember, may be inhibited by this type of drugs, which is known as sulfonylurea, and here are 2 examples. Deficiencies in acyl-CoA dehydrogenase also lead to a problem because no activation would occur, and actually every enzyme is specific for a particular chain of a particular length. This means that Very long chain fatty acids have a particular dehydrogenase. Long chain fatty acids have another enzyme. Medium chain ones, short chain ones do have the same thing. Symptoms include coma, lethargy, and the treatment includes avoid or prolonged fasting, because now we cannot activate the fatty acids and so cannot be oxidized. So please do not fast, because fasting would deplete the glycogen stores and would make the body dependent on the glucose. Because of the fasting, once the glucose or glycogen storage are depleted, we are going to use the fats. And because of the deficiency here in acyl-CoA dehydrogenase, we cannot oxidize the fat, and so here is a problem. No energy is generated, and this may lead to coma and death because there is no ATP. Finally, what if a deficiency occurred inside the alpha oxidizing enzyme? Well, here was talking about the beta oxidation problems. Now we are going to talk about the alpha oxidizing enzyme deficiency. Remember, the alpha oxidation was special for phthanic acid, and so if the phthanic acid is not oxidized, it would accumulate, and its accumulation results in retina pigmentation, nerve fibers, neuropathy or peripheral nerve fibers problems, and nerve deafness particularly. So if someone having alpha oxidizing enzyme deficient, they need to avoid dairy products or meat products. I hope this lecture helped you to understand how we oxidize fatty acids. And we have reached the end of this lecture. Thank you.